Welcome to Life Sciences by Tumi Selani. This is a YouTube online teaching platform dedicated to teaching life sciences content, sharing skills and insight to maximize your potential at academic success. Be sure to like, comment, and subscribe. And please do share the link to this channel. This will help the channel grow and reach a wider audience of learners who may appreciate the lessons and also reach their academic success. Teachers, parents, and any other stakeholder are welcome to partake in the videos and contribute to the content in the comments. Without wasting any of your time, let's get right into it. My name is Tumi Selani. I am a teacher at Milner High School. This is going to be a lesson series broken down into different lessons. For lesson 1, under the topic of the DNA code of life, we will be looking at the revision of the structure of the cell. This content you have learned about in grade 10. Let's now look into the breakdown of the lesson series. Now for the breakdown of the lesson series. The topic DNA code of life will be broken down. I've decided to break it down into 7 lessons. Each lesson is going to comprise of its own video. So be sure to hit the notification bell to be notified when I post the next video. On today's lesson, which is lesson 1, we will be looking into the revision of the structure of the cell, like I said on the previous slides. This content you learned about in grade 10. Lesson 2, which will be on a separate video, will be on the DNA structure. The following lesson, on a separate video of course, will be DNA replication. Lesson 4 is going to look into the history of the discovery of DNA. Lesson 5, we will look at RNA structure. Lesson 6 will be on protein synthesis. It will be broken down into 2 parts. Lesson 6 will be the process of protein synthesis, which comprises of transcription. It's supposed to be transcription there. I've made an error. Excuse me for the error. Transcription, movement of mRNA, as well as translation. The second part of lesson 6 will be based on amino acid coding. On a separate, and the last video on the lesson series will be lesson 7, which will be on DNA profiling. There we are going to look at the definition and uses of DNA profiling, factors that contribute towards the reliability of DNA profiling, interpretation of DNA profiles, where we will be determining paternity as well as determining a possible criminal using the technique of DNA profiling. What we will be looking at in this lesson, we will be looking at the structure of the basic unit of life, which is the cell. All right. Now, we are now going to look at the different organelles and other structures of the cell, and we will be looking at the functions of these organelles and structures. Remember, we will not be looking at every single organelle of the cell. We will not be looking at every single part of the cell. We will only be focusing on parts of the cell that are relevant for the grade 12 curriculum. Okay, basic structure of the cell. Now, the cell, like I said, is made up of different components. When we talk about a component, we are talking about different parts. Mainly, we have the structures that surround the cell, okay? The structures that surround the cell, depending on which type of cell, from which organism. From grade 10, we know that plant cells have a cell wall. And then the cell wall is tough and it is made up of cellulose. And it offers protection, mainly protection for the cell. And then All cells, including plant and animal cells, are made up of cell membranes. Okay? Animal cells only have the cell membrane surrounding it, but plant cells have the first layer, which the outside layer, which is the cell wall, and further inside it would have a cell membrane. Okay? Now, the cell membrane is there for permeability. It controls the substances entering and leaving the cell. So the main function of the cell membranes is to separate the cells from the outside environment and control all those substances that move in and out of the cell. And then the cell membrane offers some level of protection, especially for animal cells because they do not have a cell wall. The second component of cells is the cytoplasm. All right? Now, the cytoplasm is a watery jelly-like substance of the cell. What it does is that it is the site, or it is the location of many chemical reactions that take place in the cell. For example, that's where protein synthesis takes place, the process where the cell makes its own proteins. We are going to look at protein synthesis in lesson 6. The cytoplasm, it holds the inner components of the cell, such as the organelles. So it is where the organelles are, quote unquote, floating. And the last component of the cell, we have the organelles. Now, when we talk about the organelles, what are we talking about? We are talking about specialized structures within the cell. What do? They perform specific functions for the cell to make sure that the cell functions at its optimum. So the word organelle means a small organ. Think about the different organs of the cell, of your body. The different organs are the brain, an eye is an organ, the liver, the kidney, the stomach, the skin is an organ as well. So what organs do, they perform different functions for the body to make sure that the body is working efficiently and the body is working effectively. Okay, so organelles are microscopic, very small organs that are found inside the cell. So each organelle performs like an organ. It has a specialized function, and the organelle will perform this function for the overall functioning and survival of the cell. So the three components of the cell are the surrounding structures. Plant cells would have the cell wall and the cell membrane. Animal cells will only have the cell membrane. Inside the cell membrane, we would have a jelly-like watery substance called the cytoplasm. That's where a majority of the chemical reactions take place. And we have organelles, which are essentially living structures of the cell that have specialized function to make sure that the overall cell functions well. Continuing with the basic structure of the cell, we will be looking at some visuals and trying to identify the different parts and organelles of the cell. So here we have an animal cell, and then there we have a plant cell. All right, now let's try and label the different parts of the cell, okay? If you need to pause, please do pause and comment what you think the part is. The first part is on the outside of the animal cell, but it's the second layer. If you can check, it is the second layer of the animal cell. Okay, now which part did we say surrounds the cell, but it's found in both the animal cell and the plant cell? That part is called the cell membrane. And then we said that the cell membrane is there to control substances that move in and out of the cell. So it is mainly responsible for permeability. Which part of the cell did we say plant cells have, but animal cells don't? And this part is quite tough. It is made up of cellulose, and it offers protection for the plant cell. Still remember? That part is called the cell wall. Okay? Moving in. So these are the two surrounding structures. Moving further into the cell, we have this part and we have that part. As you can see, the label line is showing at a random place. So that means that this part is all inclusive of the inner parts of the cell. It is all in the cell. It is found everywhere within the cell. Clue, it is jelly-like. It's a semi-liquid, and that's where chemical reactions of the cell take place. And that part is known as the cytoplasm. The next part is an organelle. There is the organelle in the animal cell, and in the plant cell, there it is. The function of this part is said to be the powerhouse of the cell. So it is the mini ESCOM of the cell. It provides energy in the form of ATP for metabolic reactions of the cell. Can you still remember that from grade 11? Can you still remember that from grade 10? You did this also in grade 9. This is the mitochondrion. So one singular mitochondrion, many mitochondria. And the function, like we said, is that the mitochondria produce energy for the cell. Energy in the form of adenosine triphosphate, abbreviated as ATP. The next organelle that we are going to look at is a very common organelle that we all know about. As you can see, it is that organelle there on the plant cell and that organelle there in the animal cell. This organelle houses or it stores the genetic material of the cell, the DNA of the cell. It is also responsible for basically instructing the cell of its functions, how it should function, how it should look. It controls the overall activities of the cell. This organelle is called the nucleus. Like we said, it stores the cell's genetic material. So if you don't want to say genetic material, you will say it stores the cell's DNA. It controls the cell's processes, overall processes. Now, the next one that is relevant for grade 12 is, okay, now when you look at the plant cell, you will see outside the nucleus, we find those little dots, those little organelles that look like granules of sand. Let's look at this side. Just outside the nucleus, we will have the endoplasmic reticulum, the ER. The ER, for you, it's not important for you to know or remember the ER for the purposes of grade 12. But just outside the nucleus, you'll realize that we have the ER, the rough ER. They call it the rough ER because it has those small little granular organelles. These organelles are responsible for protein synthesis, and these organelles are known as the ribosomes. Like I said, they are responsible for protein synthesis. This is where proteins of the cell are made. And then last but not least, we have an organelle that we find in the plant cell. However, it is not there in the animal cell. Now, this organelle is responsible for the process of photosynthesis. This is where radiant energy is going to be trapped. Glucose is going to be broken down. Nope. Excuse me. Scratch that. This organelle is where radiant energy is going to be trapped using water to produce glucose, to produce food for the cell. And that is the chloroplast. That is the chloroplast. Okay, moving on. Okay, so I have a little simulation for you here for us to look into the different parts of the cell. Excuse me. All right, let's check. Now, this is what the cell looks like in terms of the simulation from the outside. So from the outside, this is an animal cell because it does not have a cell wall. So what is the surrounding structure of the animal cell? That is the cell membrane. So what we see from the outside is the cell membrane. And then, there we go. We have the cell membrane. We are now going to be moving into the cell. And then what you will notice, let me just go back. What you will notice is that inside the cell, there is a jelly-like or liquidy-like or fluid-like substance that is found inside the overall cell, and that is known as the cytoplasm. From there, let's look at the different organelles of the cell. Let's look at the different organelles of the cell. All right, now, the organelles of the cell that are important for grade 12 are, okay, before we look into that, like we said, organelles are little organs, are small little organs. There are structures inside the cell that are responsible for specific functions for the cell to be able to function and survive. Okay, now let's check. So those organelles over there are the mitochondria, and then we say they're responsible for producing energy in the form of ATP. There we have the nucleus, okay? We have the nucleus. The nucleus is surrounded by the rough endoplasmic reticulum. Like I said, it's not important for you to know about the ER for now. In grade 12, you dealt with it in detail in grade 10. And just in the cytoplasm, we have those granular, as you can see, we have those granular organelles, those small little organelles, and they are known as the ribosomes. Continuing. Now, when we look into the nucleus of a cell, there is something that we should notice. There is something that we should notice in terms of the nucleus of the cell. If I can just get to that part, let us check. The nucleus of the cell, and these parts you should know, okay, is surrounded by its own membrane. And the membrane of the nucleus is called the nuclear membrane. Some sources refer to it as the nuclear envelope, but I'm going to refer to it as the nuclear membrane. You will see that from the outside, the nuclear membrane is not smooth at all. It's not smooth at all. Excuse me. The nuclear membrane is not smooth. It has small little holes on it. There are small little holes on it. It has small little pores on it. And these pores are known as the nuclear pores. They are pores of the nucleus, hence they are called nuclear pores. They are found on the membrane of the nucleus, which is called the nuclear membrane. Now, let's move into the nucleus and see what are we going to find inside the nucleus. So we are going to be moving into the nucleus through the nuclear pore. Okay. Like we said, the nucleus stores the cell's genetic material, or it stores DNA. So these threads that you see inside the nucleus, this is known as chromatin. okay, or a chromatin network. This is a network of the cell's DNA or a network of the cell's genetic material. Now, inside the nucleus, we have a water-like substance of the nucleus. This water-like substance is called the nuclear plasm. Not to be confused with the cytoplasm. The cytoplasm is the plasm or is the water-like substance of the overall cell, but the nuclear plasm, nuclear, is the watery substance of the nucleus itself. So the nucleus is engulfed by, not engulfed. It is comprising of the nuclear plasm. And then when we look into the nuclear plasm, we'll see floating around will be these threads of genetic material or threads of DNA that we call the chromatin network. Moving on. Okay. Now, the chromatin, we'll discuss this later on. The chromatin forms chromosomes when the cell starts to divide, but let's not discuss that for now. Let's focus on the structure itself. Let's focus on the structure of the nucleus itself. So there we have the nuclear membrane, as you can see. Excuse me. So there we have the nuclear membrane, as you can see. This nuclear membrane separates the inside of the nucleus to the outside of the nucleus. So there will be our cytoplasm and there will be our nuclear plasm. And like I said, when we look into the inside of the nucleus, we will see a network of genetic material or a network of DNA that is called chromatin. And then the nuclear membrane, like we said, has small little holes on it that are called the nucleopores. Let's move on to the next slide. Okay, the organelles of the cell that you need to know about briefly are the nucleus, the mitochondria, the ribosomes, and the chloroplast in plant cells. Okay, all other parts are not relevant for the grade 12 curriculum. All right, now a detailed study of the nucleus. Plant and animal cells have nuclei. Now the word nuclei is plural for nucleus. We have one nucleus, many nuclei. Now the plant and animal cells have nuclei inside their cytoplasm. The main functions of the nucleus is, like we said in the past, to control all processes and chemical reactions that take place in the cell. The nucleus also contains the cell's genetic material, which is organized into two long molecules called DNA molecules. So in lesson 2, we will look at what DNA is, what does it do. So these are the two main functions for the nucleus. Now, let's have a look at the following diagram. It has, so here we have a micrograph, okay? This is a micrograph. A micrograph is a photograph of microscopic images. So this is what a nucleus looks like under a microscope. So magnified under a microscope, this is what the nucleus looks like. So a micrograph is a photograph that is taken of a magnification in a microscope, through a microscope rather. As you can see, the nucleus has the nuclear membrane surrounding it. And then we said that the nuclear membrane is not smooth. It contains those small little holes. These holes over here are known as the nuclear membranes. And then Inside the nucleus, we have a watery substance called the nucleoplasm. And then the nucleus houses the cell's genetic material or houses the cell's DNA. As you can see, those threads of genetic material are known as chromatin. And then inside the nucleus, we have the nucleolus, not to be confused with the nucleus. We have the nucleolus, and the nucleolus usually you will see it is a dark pigmented part. Mostly they are drawn as a dark pigmented dot or dark pigmented part. There it is known as the nucleolus. So parts of the nucleus: nuclear membrane, the nucleopores, we have the nucleoplasm, which is the water inside the nucleus. We have strands of DNA or strands of genetic material, which is known as the chromatin. And then we have the nucleolus. So you need to know how to label these different parts. Okay, continuing with the structure of the nucleus. The nucleus is structured as follows. We have the nuclear membrane surrounding the nucleus. So this is a double membrane that encloses the DNA or encloses the genetic material. All right. This nuclear membrane contains pores that are called nucleopores, or you can call them nuclear pores, through which substances pass in and out of the nucleus. So the nuclear membrane is in charge of permeability. It is in charge of the substances. It controls the substances that move into the nucleus and out of the nucleus. Okay, so substances, when they move in and out of the nucleus, they move through the nucleopores that are found in or on the nuclear membrane. Okay, and then we have the nucleoplasm. The nucleoplasm, like we said, is a gel-like fluid that controls metabolic activities that take place in the, not control rather, but that house metabolic activities that takes place in the nucleus. Note this, very important, this is a mistake that a lot of learners do. The mistake, the nucleoplasm and the cytoplasm. Please mind the prefix. Cytoplasm, it's a watery substance of the cell. Okay, so cyto is a prefix referring to the cell, and then nucleo is the prefix referring to the nucleus. So the plasm of the nucleus is the nucleoplasm, and the plasm of the cyto, that doesn't make sense, but you know what I mean, is the plasm of the cell. Okay, so cyto for cell, and then nucleo for nucleus. So don't mistake nucleoplasm and cytoplasm. They are not the same thing. Right, now inside the nucleus, we have the nucleolus. This is often drawn as a dark dot within the cell, the cell's nucleus. So when you look into the inside of the nucleus, you will see a pigmented dark part that is usually drawn by these artists that draw for publications like textbooks, question papers, and the likes. So that is the nucleolus, okay? The nucleolus is responsible for making ribosomal RNA as well as transfer RNA. You don't need to worry about what these two things are for now. In lesson 6, you will understand what ribosomal RNA is and what transfer RNA is. So be sure to look into that video, the video of lesson 6. Just for now, you need to know that the nucleolus is where the ribosomal RNA and transfer RNA is made. Note again, very important, do not confuse the nucleus with the nucleolus, okay? The nucleolus is found inside the nucleus. These two things do not mean the same thing. So, common mistake that people do, they confuse the nucleolus, look at the spelling, for the nucleus, and look at that spelling. Okay? The nucleus also contains, let me just erase there. The nucleus also contains the cell's genetic material, or DNA, okay? So one of the main functions of the nucleus, we said that DNA, it contains DNA. So DNA contains information about inherited characteristics. What are inherited characteristics? What is heredity or inherited characteristics? Now, inherited characteristics, the word inherited means that you receive from mom and dad, okay? Now, we have different characteristics. Characteristics are what? Your eye color, your eye shape, how tall you're going to be, how short you're going to be, your foot size, the shape of your head, your blood type, your skin tone, whether you'll be dark or you'll be light, etc. So all these characteristics are stored in DNA, okay, are coded for by DNA. So we inherit our DNA from mom. 50% of our DNA is stored in the sperm of dad or dad's sperm, and 50% of our DNA is stored in mom's ovum. Now, during fertilization, the DNA of mom and DNA of dad come together to make us. That's why 50% of our characteristics are of mom and 50% of our characteristics are of dad. The reason is that these characteristics, or the codes for these characteristics, are stored in DNA. And the DNA of mom, we inherit 50% of, and the DNA of dad, we inherit 50% of. The DNA of mom, like I said, stored in her ovum or egg cell, 50%, and then 50% of our DNA containing 50% of the characteristics is contained in dad's DNA, which is found in his sperm cells. During fertilization, they come together and they make us. All right. Now, when we look at the inside of the nucleus of each cell, we will find genetic material, or we will find DNA. Okay. Right. Moving along. So like I said, let me just erase there. Like I said, inherited characteristics or heredity. These are characteristics that are passed from parents to their offspring. All right. Depending on whether or not the cell is dividing, the DNA within the nucleus can be seen packaged in one of two ways. Okay. Now, when the cell is not dividing, when we examine the nucleus of the cell, we will see the chromatin network. So the DNA will be organized or will be packaged as chromatin network. Okay. So when we observe the nucleus and we see threads that are the chromatin network, we know that cell is not yet dividing, either by mitosis or by meiosis. And when the cell is dividing, the genetic material will be packaged in structures that are called chromosomes. Okay? They will be packaged in structures called chromosomes. Now, when we observe the nucleus of a cell and we see structures that are called chromosomes, we know that that cell is now dividing. All right. Okay, now let's see how DNA is packaged. Look at this cell. When we study this cell, what do we see? Let's check. Now, we see a chromatin network. We see threads of DNA or threads of genetic material. What does this mean? This means that the cell is not dividing. All right? However, when we observe this cell over here, instead of seeing the chromatin network, what do we see? We see structures that are called chromosomes. So when the cell is dividing, the cell's genetic material or DNA will be packaged in structures that are called chromosomes. And then when we see chromosomes, we know that the cell is either it's about to or it is undergoing cell division. Okay, let's go back to that simulation of ours. Right? Now, here is our nucleus. Like we said, the nucleus is not smooth. It has small little holes or pores in it called nucleopores. When we move inside the nucleus, when we move inside the nucleus, like we said, we will see the cell's genetic material in threads. And these threads are known as chromatin. All right? And then, like we said, that when the cell is not dividing, the chromatin or the DNA will be packaged or organized as chromatin network. Okay, so when we see this, it means that the cell is not dividing yet. It's not undergoing cell division. But let us see, when the cell is dividing, this chromatin will start condensing to form structures that are called chromosomes. So when you see chromosomes in the cell, you will know that the cell is ready to divide or is undergoing cell division. Okay, guys? All right. DNA structure, we are continuing. Now we are particularly looking into the structure of a chromosome. We want to know what chromosomes are, and what they look like. Okay, now here we are going to look at what a chromosome is. We are going to study what a chromosome is and the structure of a chromosome. Question number 1. What is a chromosome? For 2 marks. The definition. A chromosome is a structure made up of 2 components. It is made up of DNA and it is made up of proteins called histones. Okay? So a chromosome is a structure made up of 2 things. It's made up of DNA and it is also made up of histone proteins. Following question. When in the cell's life cycle are chromosomes visible in the nucleus? When in the cell's life cycle are chromosomes visible in the nucleus? And the answer there is, when the cell is not dividing, DNA is organized as the chromatin network. Actually, that is not the answer. Chromosomes are visible in the nucleus of a cell during cell division. So that's where the answer is, all right? So chromosomes are visible during cell division. We have 2 types of cell division. In grade 10, you learned about mitosis, okay? When one cell divides into 2 genetically identical cells. You learned about that in grade 10. Now, in the following topic, for grade 12, we are going to, excuse me, my throat is a bit dry. Like I said, meiosis, we are going to learn about in the following topic. Okay, that's actually the following topic, following DNA Code of Life here in grade 12. So during cell division, that's where we are able to see chromosomes. That's when they are visible in the nucleus of a cell. The following question: describe the structure of a replicated chromosome. Or they can ask this question as, how does DNA form a replicated chromosome? Okay, so the term replicated chromosome, don't worry about the word replicated. We'll clarify what a replicated chromosome is later on. But for now, let's just say that we are describing the structure of a chromosome. Okay, or they can ask this question in a different way. How does DNA form a chromosome? The answer to that is DNA coils itself around histone proteins, one tick, to make a chromatid. One tick. Two chromatids are joined together by a structure called a centromere, one tick, to make a replicated chromosome. All right, now let's check. So there, a chromosome looks like that. A replicated chromosome looks like that. Right? Now, when we look at the structure of a replicated chromosome, we will notice that it has one chromatid on the left and it has another chromatid on the right. So this will be chromatid 1 on the left and then chromatid 2 on the right. And these two chromatids are joined together by that structure over there, which is called a centromere. Okay. Now, what makes up one chromatid of a chromosome? Let us analyze. Now, a chromosome, from the definition up there, is a structure that is made up of DNA and histone proteins. Now, how are these two components of a chromosome? How are they organized? We said that DNA calls itself around histone proteins. When we study this, there is our DNA. As you can see, it is quite long. It's a very long molecule. And then there are our histone proteins. So what DNA does is that it coils itself. It wraps around these histone proteins. It coils and recoils and recoils and recoils and recoils over and over and over and over and over again to form one chromatid. So this would be one chromatid. Okay? And if we have two chromatids, as you can see over there, that is joined together by, by a structure called the centromere. So there would be a centromere that will form a replicated chromosome. So a replicated chromosome essentially is a chromosome with two chromatids, chromatid 1 and chromatid 2, that is joined together by a centromere. All right? So to repeat, a chromosome is made up of two components, DNA and histone proteins. So how is DNA organized within a chromosome? So the DNA that you see over there will coil itself or wrap itself around these histone proteins again and again and again and again and again and again and again to form two chromatids. And these two chromatids are joined together by a structure that is called the centromere. And this forms a replicated chromosome. Replicated chromosome has two chromatids that are joined together by a centromere. So like we said, when you analyze the nucleus of a cell and you see these structures called chromosomes, that indicates that it's either the cell is just about to divide or it is undergoing cell division. Okay, let us move to the next slide, where we are going to look at a simulation of this that we have been describing. Okay, on this slide, I actually have a video simulation again. Guys, remember, I do not own any rights to the videos, the clips, the drawings. Hence, I've attached the sources from which I got the videos. All right, so over there we have the cell. And then we know that from the outside, we move to the inside organelles. Same simulation. We move into the nucleus. We say when the cell is not dividing, the DNA is in threads. These threads are called chromatin network. Okay? And then, when did we say we see chromosomes again in the cell? We said we see chromosomes in the cell when the cell is dividing. So the chromatin network will start to condense, and the genetic material will be packaged in the form of chromosomes. So there, the chromatin network is forming chromosomes. So that means that the cell now is going to divide. Okay, that was question number 2 of the previous slide. Okay, now here we are looking at a structure of a replicated chromosome. Just to remind you, we said that histone proteins DNA coils around histone proteins to make one chromatid. So when you look at this chromosome, it has one chromatid, it has the second chromatid there, and then at the center there, we have the centromere. Okay, so the two components of a chromosome essentially is DNA. So a chromosome is a packaging of DNA when the cell is dividing. Okay? Now, in reverse, if we uncoil this chromatid, what do we expect to find? If we uncoil, debundle this chromatid, we expect to find two things. We expect to find a string of DNA or a long DNA molecule, and we are expecting to see histone proteins. Okay, is that what we're going to find? Let's just find out. Let's check. So we are uncoiling the chromatid, and that is exactly what we are going to find. As you can see, there is the string of DNA, and then it had coiled itself around the histone protein and coiled itself again and again and again and again and again and again to form a chromatid, and then two chromatids joined together by a centromere forms a replicated chromosome. So there is our DNA that it has coiled itself around histone proteins. So if we uncoil a chromosome, we expect to find DNA and histone proteins, because those are the two components of a chromosome. There we go. Moving on. Okay, guys, we have reached the end of the lesson on the revision of the structure of the cell. Remember, We did not look at every organelle. We did not look at every part of the cell. We just looked at revised rather, the structures of the cell that are important for the grade 12 curriculum. I cannot just leave you. They are in the following slides. We are going to be looking into questions, assessment questions. Right, so this side, we will be having our questions, and then the memorandum will be this side. All right, let's check the questions. Study the diagram of a cell below and answer the questions that follow. Now, we are given a diagram there, all right? And the instruction says, study the diagram of a cell below and answer the questions that follow. I'm going to share a very important key to answering, a very important tip, rather, in answering questions for the exam. And this skill you should practice as you're doing your informal activities in class or you're working on previous question papers. Please, in this order, you study the diagram first. And after studying the diagram, then you look at the questions. Do not look at the questions first and go back to looking at the diagram. So you should grab your pencil. If you have this in a form of a question paper, you should grab your pencil or your pen, whatever that you use to answer, and then you should study the diagram. How do you study the diagram? You analyze, you check what the diagram is, and then if you have labels, if you have structures that are unlabeled, A, B, C, D, E, F, organelle 1, you should fill them out first. That's how you study the diagram. And then you are definitely going to get questions on these unlabeled structures. Examiners will never include unlabeled structures if they are not going to ask about them directly or indirectly. Okay? Now, let's check. We have A. A is just outside of the nucleus, and it is small little dots or small little granules that is just outside the nucleus. So I submit that A is represents the I'm waiting for you to answer. Okay, A represents the organelles that are called ribosomes. Okay, so the vacuole has already been labeled, the cell membrane has already been labeled, and then we have B, C, and D. Now, B, C, and D, according to this diagram, form an organelle called organelle 1. All right, let's look. Now, organelle 1 is inside this. Organelle 1 is this structure that you see over there. Okay, now what is that? This to me looks like a nucleus. Why? There's the nuclear membrane, and the nuclear membrane has nucleopores. Now inside the nucleus, we know that we have this pigmented dot that we see over there. So B, I submit that it is, for now, I submit that it is the nucleolus. C is surrounding the nucleus, therefore it is the nuclear membrane. And then D, be very careful, follow the label line and check precisely where it is labeling. Okay, as you can see over there, it is, the label line is on this thread over there. So this thread, the answer, chromatin network, and we know chromatin network is the cell's genetic material, or it is DNA. Okay, now, if B is the nucleolus, C is the nuclear membrane, and D is DNA, B, C, and D make up organelle 1. So I submit that organelle 1 is the nucleus. Then we go to E. We go to E. When we check E, it is the label line is indicating a random void in the cell. So I submit that is the cytoplasm. And then we have these two organelles over there. And then I submit that they cannot be the chloroplasts, because remember, this is an animal cell. Why do I say is an animal cell? It does not have a cell wall. Therefore, these cannot be a chloroplast. What could they be? These are the mitochondria. Okay. Now, question 1.1 says, identify the fluid. Identify the fluid. Now, when you are reading these questions, be sure to underline the clues. All right? Be sure to underline the clues. So the clue is fluid labeled E. So it means that E here is a fluid. So this fluid is the fluid that is contained within the overall cell, right? So it cannot be the nucleoplasm. Remember, the nucleoplasm is inside the nucleus, plasm of the nucleus. So the cytoplasm is the is the plasm of the cell. Hence, cytoplasm, 1 mark. Okay, so the question said fluid labeled E. Therefore, it cannot be an organelle, cannot be a structure, cannot be a molecule. It could only be a fluid. So these clues, what they do is that they filter everything down to give you a clue as to what the answer is. So whatever answer that you give should be a fluid. You shouldn't be too far off. Following question. Let me just erase that. Okay. The following question, which organelle? Remember, we are underlining the clues. Which organelle is made up by parts B, C, and D? Now, do you see why it was important for us to study the diagram first? So, you would have written here, nucleus. So, all that you need to do is to transcribe what you had already written on your question paper to your answer sheet. So, the correct answer there is nucleus for 1 mark. The following question, provide the letter /S, letter or letters, and name or names of the part that. Be very careful, guys, of this type of question. A lot of people don't acknowledge or see this. So the answer to the question may either be a letter or it might be letters. So if it's one letter, therefore it will be one name. So 1 letter, 1 mark, 1 name, another mark. If it is letters, it would be 2 letters or 3 letters and 2 corresponding names. Okay, so please be very careful. Don't just glance and just selectively see letter and you ignore the name. Okay, so either it's going to be 1 letter or 2 letters, or 1 name and 2 names. All right. Okay, let us read the instruction again. The instruction says, provide the letter or letters and name or names of the part that. Subquestion A provides energy for the cell. Now, remember I said that examiners will either ask you directly for you to label an unlabeled part, such as here, identify fluid labeled E. So that is a direct question. It is a direct way of asking what is fluid E. However, indirectly, they have given you a function. what you have to do is identify the relevant part of the cell in terms of its letter and its name. So, like I said, examiners will never leave out unlabeled structures if they are not going to ask about them either directly or indirectly. We've seen in question 1.1 of a direct question. Now here, this is an indirect question. Okay, so sub-question A of question 1.3 is asking provide the part that provides energy for the cell. Remember, you have to give the letter or letters and name or names. Now, had you labeled mitochondria here, you would know that if the letter 1 mark, and then you write mitochondria 1 mark. Let's see if that's the correct answer. Yes. F 1 mark, the letter, and mitochondria another mark for the name. There is no other organelle that provides energy for the cell here. Therefore, it is only 1 letter and 1 name. Sub-question B. Synthesizes rRNA, ribosomal RNA. Okay, marker location is 2, therefore it's going to be 1 mark for the letter and 1 mark for the name. So rRNA is manufactured by B, the nucleolus, not the nucleus, B, the nucleolus. Let's see if that's the correct answer. Definitely is. B, 1 mark. Nucleolus, 1 mark. Please mind the spelling. C is the site of protein synthesis. The word site means location. All right? So, where does protein synthesis take place? It takes place at A, ribosomes. Let us check if that is correct. That is correct. A, 1 mark. Ribosomes, 1 mark. Now let us look at sub-question D. Contains DNA for 4 marks. Contains DNA. Now, this should raise eyebrows. This 4 marks should raise eyebrows. It should be an indicator that it is not just 1 letter and 1 name. Therefore, it's not only 1 place where DNA is contained. Because the marker location is significantly higher than the previous ones, therefore it means there's more than 1 place where DNA is contained. Okay? So, DNA is contained where? DNA is contained firstly in the nucleus. That's where we know most of the cell's DNA is contained. All right? So, okay. So it will be, I know it is not a question. I mean, it is not a letter. It's a phrase. So in organelle 1, and organelle 1 we said is the nucleus. For 1 mark for organelle 1, 1 mark for the name nucleus. Okay. And then DNA is also found, as we will see in the next lesson. DNA is also found in structure F, which is the mitochondria. Let us check. F, mitochondria, 1 mark, 1 mark. And in organelle 1, which is the nucleus. All right. Following question. Or, okay, had the question said organelle here, had it said organelle, nucleus, organelle 1, and F, mitochondria, would be the only answer. So, because they said part, it makes the answers a bit more open. So one could actually say D is a chromatin network. It's a network of genetic material, network of DNA. So D as a concession may be the correct answer as well. Following question. State 2 reasons why the cell above is not a plant cell. Is not a plant cell. We know that plant cells have cell walls, ne? And then also we know that plant cells have chloroplasts. This cell does not have both. Therefore, if it does not have both, it means that it is an animal cell. Okay, so what is the correct answer to this question? It has no cell wall and it has no chloroplasts. Okay, the question said state and not explain. So it just said state and it said 2 reasons. So there are our 2 reasons. Do we have another question? Yes, we do. The following question says, describe. Okay? Describe. Describe how molecule D, we said it's DNA or genetic material, would otherwise be arranged if the cell above was dividing. This cell is not dividing. Why? We said when the cell is not dividing, what are you going to identify in its nucleus? When you examine the nucleus, its genetic material will be arranged as threads called the chromatin network. However, if the cell is dividing, this chromatin network will condense to form structures that are called chromosomes. So we would have seen chromosomes within the cell, and we would not, the nucleus, we would not see the chromatin network. Let's read the question again. Describe how molecule D would otherwise be arranged if the cell above was dividing. The correct answer is DNA or genetic material would be packaged into 2 chromatids joined together by a centromere to form a replicated chromosome. So even though you do not write replicated, it's fine. It is in brackets, meaning this word is an option. So to form a chromosome. Okay. So chromosomes would have been seen, and a chromosome is when DNA is coiled around histone proteins to make a chromatid, and 2 chromatids joined by a centromere forms a replicated chromosome. Okay, guys, we have officially reached the conclusion of the first lesson of the DNA Code of Life, lesson 1, which was based on the revision of the structures of the cell. Be sure to like, comment, subscribe, and please do share the link to this video. It is going to help the channel a lot. It would mean that more and more people will gain access to my lessons, and it will grow the channel. It will be highly appreciated. Again, thank you so very much for watching. The next lesson will be on the DNA structure. That will be lesson number 2 on a separate video. Please hit the notification bell so that you'll be notified when I post this next video. I will try to post it in the next week so that we are up to date with our lessons and we are good and ready in class when the teacher starts with this topic. Thank you so very much. See you next time. Bye.